Hello everyone, my name is Toju Etiwe and I am a junior biology major here at Oglethorpe University. Today, I will be discussing the use of an IL-1 antagonist to prevent tendinopathy in a rat model. So for this study, I will first discuss some background information regarding what tendinopathy is, the type of receptors that help with inflammatory and anti-inflammatory responses, reasons for the study in tendinopathy, and how it affects us humans. Next, I will talk about the goal of the study, the approach of the study, the experimental design, and the results of the study. Lastly, I will discuss the conclusion of the study and some take-home points. First off, tendinopathy is the breakdown of collagen in the tendon. The tissue connecting the muscle to the bone becomes inflamed, causing a burning pain, reduced flexibility, and a reduced range of motion in a person. Tendinopathy also means a tendon has a disease or disorder. I want to make a clear distinction between tendinitis and tendinosis. Tendinitis is the basic tendon pain that stems from acute inflammation during acute micro tears. This causes pain and swelling. Tendinosis is the non-inflammatory deterioration of a tendon, meaning repetitive pain and swelling of a tendon will cause tendinosis. The reason that I chose to present the topic of inflammation of tendons is because as an athlete myself, these problems come and go, but we athletes never get to fully understand why this pain lingers during our prime years of sports. On Oglethorpe's campus, 28% of the student body participate in athletics. Within that 28%, those student athletes are susceptible to tendon injury pain. 1 to 3% of these athletes will suffer from some sort of tendon pain or injury. I have no shortage of knee problems playing both basketball and track myself. Not only do athletes suffer from tendon pain, regular students at Oglethorpe are susceptible to injuring a major tendon also. The most common injuries are in shoulders, elbows, wrists, knees, and heels. A few specific examples include tennis elbow, Achilles tendonitis, jumper's knee, and swimmer's elbow. With all these examples, it shows how the tendon in our bodies are susceptible to injury. So therefore, we need to discuss how to either prevent these injuries altogether or stop the progression of them. This starts with learning about what type of interactions are taking place within a tendon. There are some reasons why tendons become inflamed and cause pain and swelling. The big focus of the study was on the patellar tendon. The patellar tendon is the attachment of the bottom of the kneecap to the top of the shin bone. Within the patellar tendon region, there are a number of regulators, factors, and receptors that respond to immune and inflammatory stressors. The particular family that resides in the patellar tendon region is the interleukin-1 family. This family is responsible for regulating immune and inflammatory responses to infections. When the tendon becomes injured, interleukin-1 regulators are the first line of defense for the tendon. Sometimes in the patellar tendon, too much breakdown of collagen occurs, which leads to tendinopathy. This is where an interleukin-1 receptor antagonist comes into play. For the rest of the study, I will be referring interleukin-1 receptor antagonist as IL1RA. IL1RA is a naturally occurring competitive inhibitor of the interleukin-1 family. It can inhibit the IL-1 signaling pathway and prevent the attraction of cytokines that leads to increased inflammation. A cytokine is a signaling molecule from immune cells that promote inflammation. You do not want to have an abundance of cytokines in your patellar tendon region. There are two types of receptors/factors that are linked to tendinitis. The first receptor is the interleukin-1 beta receptor. This receptor helps white blood cells fight off infections in the tendon. Tumor necrosis factor alpha, or TNF alpha, is a factor that helps with disease infection resistance and helps cells with either programmed or premature cell death. When there is both an increase of interleukin-1 beta and TNF alpha, the shielding that protects the tendon becomes more stressed and breakdown of collagen, leading to an increase in cytokines, ultimately leading to the deterioration of the tendon. With all this knowledge presented, we could take a look at the background of the experiment. The study I will be discussing will determine how IL1RA causes inhibition of IL1 in rat models. Rat models are important because they are very similar to humans in that they resemble humans anatomically, physiologically, and genetically, so we are able to make a lot of connections using them. Rat models can be important in discovering how inflammatory cytokines and carrageenan can induce tendinopathy. This leads to conclusions that can almost translate to humans and can be helpful in determining if the new techniques are effective and safe for usage. These rat models are used to help and understand and examine the inhibition of IL-1 and IL-1RA in patellar tendons and how IL-1RA would prevent acute tendinitis changes in rat patellar tendons. In this rat model experiment, scientists induced carrageenan to see if tendinitis would occur, and if it did, IL-1RA was added to inhibit IL-1 and decrease the disease effects of tendinopathy. The purpose of the study is that there was to evaluate the inhibition of IL-1 beta activity with an IL-1 receptor antagonist and how that IL-1RA can effectively inhibit several types of changes seen with tendinitis. The experimental question of the study asks, is there any way to prevent and decline pathologic tissue changes that are associated with tendinitis? While the scientists of the study hypothesized that IL-1RA will prevent histological, mechanical, and structural pathologic tissue changes that are caused by induced tendinitis. For the approach, IL-1 was injected and tested in Sprague-Dawley femur rats. For the experimental design, scientists of the study first obtained 36 Sprague-Dawley retired breeder rats given from the Institutional Animal Care and Use Committee. Next, the 36 rats were put into 3 groups, 12 in each group, and they were named a control group, a carrageenan-only group, and an IL-1RA group. They were injected once a week for 3 weeks. Before the rats were injected, they were given anesthesia. Both patellar tendons of the rats were injected also. The control group con was only injected with phosphate buffer saline to help the tissues cells from not keeping a constant pH to keeping a constant pH and keeping those cells from rupturing. The carrageenan group was injected with 100 microliters of 2% carrageenan in PBS. Carrageenan is an additive that causes inflammation when injected into a tendon region. Lastly, the IL-1RA group was injected with 100 microliters of 2% carrageenan in PBS with 0.94 microliters of IL-1RA and PBS. Each injection was made fresh, weekly, and sterilized using an autoclave after each injection. After each injection, the rats were monitored at a 12-hour dark light cycle for 3 weeks. When the study was complete, the rats were euthanized and lateral radiographs were taken of the right patellar tendon. These lateral radiographs are X-ray photos of the rat's right patellar tendon region. It is also an in-depth analysis of the patella and major ligaments in the rat's right knee. Lastly, the left limbs of the rats were harvested for histological evaluations, and the remaining right limbs were harvested for 3 different evaluations that include IL-1 beta levels, collagenase activity, mechanical properties of the patellar tendon region. The first evaluation taken were for IL-1 beta levels. In this evaluation, the scientists used an enzyme-linked immunoabsorbent assay to examine IL-1 beta levels. If there are more signs of IL-1 beta, that means there is an increased inflammation that was occurring, meaning that cytokines are being overproduced in the patellar tendon region. Next, histological evaluations were taken. Histology is the study of microscopic structure of a tissue. In this study, scientists had 12 regions of interest with 5 parameters, including inflammatory cell presence, fiber arrangement, nuclear rounding, angiogenesis, and cell density. Each region was scored from 0 to 3. A 0 score meant the tendon was fine and normal. A 3 meant the tendon was inflated and there was problems with it. For mechanical testing, stiffness and ultimate load was calculated on the patellar tendon region. Stiffness is how much the patellar tendon can move and how tight it feels. Ultimate load is how much the patellar tendon could handle, such as weight, pressure, and other external factors being applied to that region. Lastly, collagenase activity was evaluated. This was a measure of bacterial collagenase and the screening and characterization of collagenase inhibitors. Scientists scanned for receptors, factors, and inhibitors that either negatively or positively affected the collagen in the patellar tendon region. Figure A and B show the patellar tendon groups. The patellar tendon is the white, glossy area that is seen within all the figures. Figure B shows a greater difference in the glossy white patellar tendon region. What is not clearly seen in the figures is in the control group, the tendons were clearer, thinner, and had a less thick area of sclerosis, which is the hardening of tissue. This indicates that there is more fixing of the patellar tendon. Figure A shows a better example of how the IL1RA group's patellar tendon is also extremely clear, thin, and is extremely has less thick areas of sclerosis in the patellar tendon region. Same could be said for the control group in A also. This indicates that in both the control group and the IL1RA group, there were no increased levels of IL-1 collagenase activity, leading to the inflammation of the tendon. Also, within figure A and B, we can see clear distinct features of the carrageenan group. One feature of this group is that it was noted by scientists that in the study, the tendon was shortened at the top of the patellar tendon region. And was shorter than the other 2 groups, meaning there was damage done by the the carrageenan that was injected. Figure B shows that in the carrageenan groups that the redness of the patellar tendon shows an increased inflammation. The red area in the patellar tendon happens because there's rapid cell growth producing new tissues, which is called proliferate. This area has also suffered from a hypervascular response, which is an increased number of concentration of blood vessels surrounding the tendon, seen more in figure B than A. In this figure, the left limbs of the rats were taken and the patellar tendon region, which is at the top, was stained. There were visual and statistical differences found regarding to inflammatory cell infiltration, angiogenesis, and cell density when the carrageenan group was compared to either the control or IL1RA group. The carrageenan group is also called the inflamed group, while the IL1RA group is also called the treated group. The black dots that you see at the top of the page show cell infiltration. This is if cells stay in the top of the region of the patellar tendon or move down the limb, causing proliferate. In the inflamed group, we see the black dots or cells have infiltrated the lower parts of the limb, spreading down the sides, causing proliferate in the tissue. In the top region of the image, we can also see how the cells are clumped together in circles. Cell density is cells per unit volume, and in the top of the photo, we see that both the control and treated group both have great cell density because their cells are where they're supposed to be, in which is the patellar tendon region. The inflamed group has some cells in the patellar tendon region, but we can see that the density is not great because some of those cells have moved away from the patellar tendon region to the lower parts of the limb. Lastly, angiogenesis, which are new blood cells created from old blood vessels that are able to give oxygen and nutrients to the tissues in the patellar tendon region. The reddish lines indicate that the new blood vessels ready to carry oxygen and nutrients to the upper part of the patellar tendon region. As you can see, the inflamed group is darker because the cell infiltration is causing the blood vessels to degrade and have slices in between them. In the two other groups, the blood vessels look very healthy and readily supplying needs like oxygen and nutrients to the upper parts of the patellar tendon region. There are clear and distinct differences that can be seen in both figures showing the effects of induced inflammation and increased IL-1 levels. A few points that you should remember the next time either you or someone you know is dealing with or talking about tendon problems. Tendinitis is the basic pain and swelling of a tendon. Tendinosis is the deterioration of a tendon because of repetitive swelling. And lastly, tendinopathy is the disease that causes the breakdown of collagen in the tendon. This leads to scientists figuring out that if there is a blockage of IL-1, that means IL-1RA is able to prevent downstream production of cytokines that leads to inflammation. If there is inhibition of IL-1, then there was a decrease in tendinopathy in the IL-1RA animals, concluding that IL-1RA has the ability to prevent this disease that causes the breakdown of collagen in an organism's tendon. But even though these breakthroughs have been done in different animal models, including rats, rabbits, horses, and mules, it is hard to predict tendinopathy and difficult to translate these results to clinical trials in human tissues, which leads scientists to believe that IL-1RA may be a viable option for tendinopathy, tendinitis, and tendinosis, Before using IL-1RA in human trials and further studies in different animal models, we need to fully determine if this is the right treatment that we need to use in humans. Here are my references, and I hope you enjoyed my talk. Thank you.